In this module, we are going to discuss the first unit of paper 2, History and Development of Microbiology. In this first lecture, we will talk about the definition of microbiology, the scope of microbiology as a subject, the early contributions of Robert Hooke and Anton van Leeuwenhoek, and the controversy associated with spontaneous generation theory. Microbiology is the study of living organisms that are too small, even less than a millimeter in diameter, and therefore cannot be seen by the naked eye. These organisms of microscopic size include bacteria, fungi, algae, protozoa, and also viruses. Microbiology deals with their form, structure, reproduction, physiology, metabolism, and classification. What does a microbiologist do? A microbiologist usually first isolates a specific microorganism from a population, cultures it, and then studies its structure and processes. Thus, microbiology employs techniques such as sterilization and the use of culture media and microscopy that are necessary for successful isolation and growth of microorganisms. Thus, we can define microbiology as the study of microorganisms and also the techniques required to study them. Microbiologists study microbiology as two major disciplines. The first is basic microbiology. As a basic biological science, microbiology provides some of the most easily available research tools for studying the various life processes. Microbial cells have many biochemical properties in common with the cells of multicellular organisms. Microbial cells can grow to extremely high densities in laboratory cultures and are readily amenable to biochemical and genetic study and hence make excellent models for understanding cell functions in higher organisms. The second major discipline is applied microbiology. As an applied biological science, microbiology deals with many important practical problems in medicine, agriculture, and industry. Some of the most important diseases of humans, plants, and animals are caused by microbes. They also play a major role in soil fertility and many large-scale industrial processes such as production of antibiotics, vaccines, and enzymes. The combination of genetic engineering and microbiology has even given rise to a whole new discipline, biotechnology. Some other branches of applied microbiology are medical microbiology. It deals with the diseases of humans and animals. Medical microbiologists identify the causative agent of the disease and plan measures to eliminate it. Public health microbiology. It is closely related to medical microbiology. It deals with the control of the spread of communicable diseases. Industrial microbiology. It is concerned with the use of microorganisms to make valuable products like antibiotics, vaccines, steroids, alcohols, vitamins, amino acids, and enzymes. Environmental microbiology. It is concerned with the relationships between microorganisms and the living and the non-living habitats. Environmental microbiologists study the contributions of microorganisms to the biogeochemical cycles like carbon cycle, nitrogen cycle, phosphorus cycle, and sulfur cycle. Food and dairy microbiology. It is concerned with the prevention of microbial spoilage and the transmission of foodborne diseases. It also deals with the use of microorganisms to make food such as cheese, pickles, and beverages. Agricultural microbiology. This field is concerned with the impact of microorganisms on agriculture. An agricultural microbiologist tries to combat plant diseases that attack important food crops and works on methods to increase soil fertility and crop yields. We will now discuss the early contributions made in the field of microbiology by some scientists. Robert Hooke was an English natural philosopher and was one of the most inventive and ingenious minds in the history of science. In 1660, he, along with Robert Boyle and a few other members, formed a scientific society which, in 1662, received a charter from King Charles II and became the Royal Society of London for the communication and publication of scientific work. As a curator of this society, he developed a primitive microscope with a monoocular eyepiece, a wooden tube or barrel, a stage to hold a specimen, and a glass globe full of water to concentrate light onto it. These microscopes gave a 25 times magnification. Robert Hooke was the first person to observe cells when he studied thin slices of cork, as can be seen in the picture. Let me take the privilege of quoting Robert Hooke from his own famous book, Micrographia. I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of it were not regular. These pores, or cells, were indeed the first microscopical pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that had made any mention of them before this. Thus, it was Robert Hooke who coined the term cell. In 1665, the Royal Society published his Micrographia, which was an accurate and detailed record of his observations, illustrated with magnificent hand drawings. As mentioned in the previous slide, Robert Hooke illustrated the cork cells that he had observed. Hooke was the first person to describe and draw a microorganism, a mold he found growing on the sheepskin cover of a book, a head louse, and a flea, and many such specimens. Micrographia represents one of the most important books in science history because it awakened the learned and general population of Europe to the world of the very small, revolutionized the art of scientific investigation, and showed that the microscope was an important tool for unlocking the secrets of nature. The first person to observe and describe microorganisms was Anton van Leeuwenhoek. What Galileo, Newton, and Einstein are to physics, Leeuwenhoek is to microbiology. Anton van Leeuwenhoek was a Dutch tailor, merchant, and a lens grinder. The credit for the discovery of microorganisms goes to him. Leeuwenhoek was born in Delft, the Netherlands. He had an insatiable curiosity and a desire to do everything for himself. Now, as a tailor, he needed to examine the quality of cloth. So, instead of buying a magnifying glass already available, he ground his own lenses to make microscopes with short focal length, lenses giving magnifications between 30 to 300 times. Soon, he began wondering what everything around him actually looked like. It is said that he was greatly inspired by reading Hooke's Micrographia. Leeuwenhoek's microscopes did not resemble our present-day sophisticated microscopes. As can be seen in the figure, the spherical lens was mounted between two metal plates. The specimen was placed on the point of a blunt pin attached to the back plate and was brought into focus by manipulating two screws which varied the position of the pin relative to the lens. Let us now discuss some of Leeuwenhoek's observations. Leeuwenhoek observed a variety of things including rainwater, pond water, blood, and even scrapings from his own teeth using his microscopes. One day, he observed a drop of pond water and was astonished to see small moving creatures. He called these small creatures as animalcules, which meant very small animals. He made accurate drawings of these animalcules and sent them in a series of letters to the Royal Society. Today we know these small animalcules as microorganisms. His greatest accomplishment was his discovery of the microbial world. Altogether, it is believed that he sent 190 papers to the Royal Society. In the figure on the left, you can see that A and B represent rod forms and C and D indicate the pathway of the motion. That is, he depicted how a bacterium is moving from one position, that is C, to another position, that is D. A spherical form is shown in E and the longer types are depicted in F. The longer type shown in figure G appears to be a spiral bacterium. Leeuwenhoek found many of these organisms in the samples which he acquired between his teeth. Leeuwenhoek reported the existence of microorganisms in 1674, but microbiology as a field of science did not develop significantly for almost 2 centuries because of two main reasons. One, Leeuwenhoek never trained an apprentice, and two, he never sold or gave away any of his microscopes. However, it is said that his daughter Maria, who was also his assistant, requested him and could convince him to donate a few microscopes. Leeuwenhoek died in 1723 at the age of 90. On his death, he passed on an ornately boxed set of 26 solid silver instruments with mounted specimens to London's Royal Society. In 1981, British scientist Brian Ford rediscovered some of Leeuwenhoek's original specimens in the archives of the Royal Society of London. The specimens were found to be of good quality and remarkably well preserved. Ford carried out observations with a range of microscopes, adding to our knowledge of Leeuwenhoek's work. For his contributions in microbiology, Antony van Leeuwenhoek is rightly called the father of bacteriology. Moving on to the next topic, that is the controversy of spontaneous generation theory. Scientists began to work